Howdy. Welcome to 2023. 1st January 2023. Science alert. Scientists grew stem cell mini brains and then the brains sort of developed eyes. Brain organoids with optic cusps at a day 60 of development. How beautiful. Yeah. Mini brains grow in a lab from stem cells spontaneously developed rudimentary eye structures. Scientists reported in a fascinating paper in 2021 on tiny human-derived brain organoids grown in dishes. Two bilaterally symmetrical optic cusps were seen to grow, mirroring the development of eye structures in human embryos. This incredible result could help us to better understand the process of eye differentiation and development, as well as eye diseases. Our work highlights the remarkable ability of brain organoids to generate primitive sensory structures that are light sensitive and harbor cell structures similar to those found in the body, said neuroscientists J. Gopalakrishnan of University Hospital Düsseldorf in Germany in a 2021 statement. Yeah, there could be some bad jokes made about this, but anyway, German doctors and stuff. These organoids can help to study brain-eye interactions during embryo development, model congenital retinal disorders, and generate patient-specific retinal cell types for personalized drug testing and transplantation therapies. Oh, how beautiful. It looks at us. But it doesn't, I don't think it blinks. Brain organoids are not true brains, as you might be thinking of them. They are small, three-dimensional structures grown from induced pluripotent stem cells, cells harvested from adult humans and reverse engineered into stem cells, that have the potential to grow into many different types of tissues. In this case, these stem cells are coaxed to grow into blobs of brain tissue without anything resembling thoughts, emotions, or consciousness. How do they know? Such mini brains are used for research purposes where using actual living brains would be impossible, or at the very least, ethically tricky. Testing drug responses, for example, or observing cell development under certain adverse conditions. This time, Gopalakrishnan and his colleagues were seeking to observe eye development. In previous research, other scientists had used embryonic stem cells to grow optic cups, the structures that develop into almost the entire globe of the eye during embryonic development. Another research had developed optic cup-like structures from induced pluripotent stem cells. Rather than grow these structures directly, Gopalakrishnan's team wanted to see if they could be grown as an integrated part of brain organoids. This would add the benefit of seeing how the two types of tissue can grow together rather than just growing optic structures in isolation. Eye development is a complex process, and understanding it could allow underpinning the molecular basis of early retinal diseases, the researchers wrote in their paper. Thus, it is crucial to study optic vesicles that are the primordium of the eye whose proximal end is attached to the forebrain, essential for proper eye formation. Previous work in the development of organoids showed evidence of retinal cells, but these did not develop optic structures, so the team changed their protocols. They didn't attempt to force the development of purely neural cells at the early stages of neural differentiation and added retinol acetate to the culture medium as an aid to eye development. Yeah, eye drops. Their carefully tended baby brains formed optic cusps as early as 30 days into development, with the structure clearly visible as 50 days. This is consistent with the timing of eye development in the human embryo, which means these organoids could be useful for studying the intricacies of this process. There are other implications too. The optic cups, I always try to say cusps, but they're cups, contained different retinal cell types, which organized into neural networks that responded to light and even contained lens and corneal tissue. Finally, the structures displayed retinal connectivity to regions of the brain issue. So they can see. Maybe they think, what's going on? Why am I in this glass here? In the mammalian brain, nerve fibers of retinal ganglion cells reach out to connect with their brain targets, and as an aspect that has never before been shown in an in vitro system. Nerve fibers of retinal ganglion cells reach out to connect with their brain targets, an aspect that has never been shown in an in vitro system. And it's reproducible. Of the 314 brain organoids the team grew, 73% developed optic cusps. The team hopes to develop strategies for keeping these structures viable and longer timescales for performing more in-depth research with huge potential, the researchers said. Optic vesicle-containing brain organoids displaying highly specialized neuronal cell types can be developed, paving the way to generate personalized organoids and retinal pigment epithelial sheets for transplantation, they wrote in the paper. We believe that these are next-generation organoids, these next-generation organoids are helping to model retinopathies that emerge from early developmental disorders. Yes. Happy New Year. Be like him or her. Keep your eyes open. Never stop learning. Thanks. Bye.